Hey guys, in a previous video we took a look at the body's innate immune system and how our body functions in a non-specific way to fight against pathogens, both blocking them from entering the body and attacking them once they are inside the body. Today we're going to take a look at the body's specific immune system. Acquired immunity is a defense against one particular disease that, as the name implies, can be acquired or developed in response to exposure to that disease. Now, this system relies on two types of cells, known as B cells and T cells. Both of them are white blood cells, but they're named based on where they develop. B cells develop in the bone marrow, and B cells generate antibodies, large proteins that function outside of the cell and circulate in the bloodstream to fight against pathogens. Now, because these antibodies are circulating outside of the cells, B cells are very important for immune response against pathogens that are extracellular, in other words, outside of our cells. Now, in contrast, T cells develop in the thymus. Now, one thing I want to be clear, when I say develop, I actually mean the process of, we're going to see that B and T cells go through a process of being generated and then being sort of maturing. We're talking about where these cells mature. All of the white blood cells are actually born in the bones, but they mature in different places. Now, T cells develop in the thymus, and they tend to fight pathogens inside the cell. And we're going to see there are two types of T cells, what we call killer T cells and helper T cells. Now, let's take a look first at B cells and how they function. Now, B cells produce antibodies. And before we start talking about antibodies, what I'd like to do is give you guys a little bit of vocabulary that's going to become important as we talk about these antibodies. Now, these two words that you're about to hear sound very, very similar. They're actually quite different. The first is an antigen. An antigen is a structure to which an antibody binds. Now, typically an antigen is a small molecule or piece of a molecule that's found on the host. The antigen is much smaller than the host itself. If my hand is the pathogen, the antigen might be my fingernail. Antigens are very specific, and they fit together with an antigen binding site on the antibody in a lock and key mechanism, very similar to how an enzyme would fit with a substrate. That specificity means that every antibody can only bind one specific antigen. And we're going to see that that's incredibly important. This is what allows antibodies in your body to recognize a pathogen as different from one of your body's own cells. It's also what creates things like incompatibility of blood types. It's what creates things like the fact that you get a new seasonal flu every year and you don't really develop immunity towards it because we have immunity towards last year's seasonal flu rather than this year's seasonal flu. Now, these antibodies are designed to fight pathogens outside the cell. And as we said, they're a very large protein molecule. And this protein molecule is made up of multiple chains joined together in what we call quaternary structure. Now, those protein chains at their very tips have antigen binding sites. The shape, as we said, is very specific to the particular antigen involved. So another thing that's important to recognize is that every single antibody has multiple antigen binding sites, not just one. And this becomes important when we take a look at how antibodies actually interfere with the growth and development of pathogens. Now, because an antibody has multiple antigen binding sites, antibodies could bind to more than one pathogen. And as a result, they can basically cover a pathogen, causing inactivation. They might just interfere with a bacterium growing or dividing. They might interfere with a virus docking with a host cell and infecting it. They can also lead to what's called agglutination. Now, in agglutination, what we simply do is bind all the pathogens together. It's kind of like so that our immune system can gobble them up. We learned about phagocytic cells and how those are fighting pathogens. If you imagine trying to eat pathogens, it's a lot easier if they're all clumped together. By joining them together with antibodies, it's much simpler for a macrophage or something like that to gobble up all the pathogens all at once. It's like trying to eat rice. If the rice sticks together, it's very easy to lift it to your mouth. If it's falling apart into individual little grains, it's a mess. By the way, as an aside, whoever came up with the idea of nonstick rice, I have my issues with that. The whole idea is that it's easier to eat if it sticks together. Same way here, if the pathogens stick together, it's much simpler for our immune system to deal with it. In addition, antibodies binding to the outside of a pathogen may target it for destruction by the complement system and other things like that. So once antibodies cover and clump together these pathogens, it makes it much easier for the rest of our immune system to fight against them. Now, B cells, once they're produced, tend to go through what we call clonal selection. Now, the trick to remember is that antibodies are specific, and indeed, every single B cell produces a different type of antibody. Inside your body, you've got millions and millions of different types of B cells. Some with antibodies against the flu that you had last year, some with antibodies against an antigen that might be found on a molecule that's never ever been created in the history of the world. Now, the way in which these B cells produce those antibodies is tremendously clever and actually won the Nobel Prize for Dr. Susumu Tonegawa and other researchers, because it turns out that these B cells have different editing of their actual genome. So we always say that every cell has the same DNA. It's not quite true in this case. It turns out that they put the antibody together with like little bits or cassettes that we say. So you take one of 1,000 A cassettes and one of 1,000 B cassettes and one of 1,000 C cassettes, and you put these together and you get a unique antibody in every single B cell. And as a result, you get 1,000 times 1,000 times 1,000. You get a large number, a billion different possible antibodies, even with just 3,000 genes. That's roughly how this works, and that process called VDJ recombination is something that's really unique to immune system cells. And the discovery that this was actually functioning in this way, that these are forming this way as they mature, that's remarkable. Now, the fact that these cells are unique and each react against a particular thing means that, well, unfortunately, we don't have a lot of any one type. So when your body encounters a pathogen, what you have to do is say, aha, we don't need a lot of B cells. We need a lot of B cells that are specific to that one pathogen. So what happens is the B cell that is activated, the B cell whose antibody is needed, clones itself. It goes through this process of clonal selection, where instead of just making more B cells randomly, we make exact identical copies of that one B cell that's necessary. That one B cell now becomes a large part of the population and can fight against that pathogen. Now, T cells are responsible for helping in that process. Helper T cells can help with the clonal selection of B cells and killer T cells. Now, killer T cells they fight against intracellular infections that are inside our host cells, by killing body cells that are displaying particular signs that they are infected. Now, the way that they do this is a really fascinating glimpse at sort of how we monitor our own cells. Every normal body cell has something called MHC, or major histocompatibility complex, on its surface. This is a molecule that basically serves like a shop window. It displays what's inside of that body cell. If you walk past a shop on the street and you want to know what's inside, even if you can't tell from the name or the sign, what you often do is simply look in the window and you can see, oh, they sell shoes, or they sell sporting goods, or they sell ballet slippers. Whatever it is, you can tell by what's on display in the window. MHC sort of works the same way. What MHC does is it grabs molecules from inside the cell and displays them on the cell surface. Now, by doing this, other cells get a good idea of what's going on inside that body cell. Now, if you're infected with a pathogen inside your cell, some of the molecules from that pathogen will get displayed on the cell surface. They'll get displayed on MHC. Now, that antigen from the pathogen gets displayed on MHC and is recognized by a killer T cell. The killer T cell then binds to the combination of MHC and the antigen and initiates apoptosis, or programmed cell death, by sending a chemical death signal to the body cell. Again, shutting it off specifically. It's very much, to continue the shop window analogy, as if you're a police officer walking down the street. The killer T cell is the police officer, walks down the street and was like, okay, great, we've got the jogging store with the running shoes and the leggings, and then we go and we've got the store selling bagels, and then, oh my goodness, this next store has like machine guns, and yeah, I don't think that seems appropriate, so we're going to shut it down. The killer T cell notices that there's something inappropriate inside the body cell and shuts it down. Now, the immune system can also go wrong. And I want to mention specifically 3 threats to the immune system that we hear about a lot. The first is HIV, or the human immunodeficiency virus. And AIDS, or acquired immune deficiency syndrome, is the set of symptoms that develop when someone suffers from HIV. Now, HIV targets cells in the immune system. And because of this, the immune system is depressed. As a result of the depression of the immune system, humans are much more susceptible to other diseases. So HIV itself is not lethal. It's the fact that it leaves the body exposed and to other types of diseases, which can cause severe problems. Now, one of the things that's in an immunocompromised individual, you not only see things like infectious disease, you also see higher incidences of cancer because our immune system is fighting against cancerous cells at all times as well. Now, another threat to the immune system are things called autoimmune diseases. Now, auto means self. So in an autoimmune disease, the body attacks its own cells. This comes down to basically a mess up in the development of the cells of the immune system. When immune system cells are produced, we talked about how the antibodies or the T cell receptors that are generated on their surface, these are produced kind of randomly. Now, during development, antibodies or T cell receptors, things like this, that react against cells from your own body, those are gotten rid of. Cells that contain those basically are killed off before they're released into your body to prevent your body from attacking itself. However, in some instances, cells get through that attack your own body cells, and these lead to autoimmune disorders. Some types of diabetes are caused by autoimmune disorders in which the body attacks some of the cells that, for example, receive insulin. Other autoimmune disorders are things like arthritis, where the body is actually causing inflammation as a result of an inappropriate immune response. Another threat to the immune system are allergies. Now, in an allergy, what we have is a reaction not to one of our own cells, but to something innocuous in the environment. Pollen, for example, comes into our body and our body treats it like a pathogen. So we trigger an inflammatory response. We trigger all of these things that are designed to fight against a pathogen, but instead we're triggering that in response to something that's really not going to cause us any harm. The only reason that an allergy is uncomfortable is because our own body is reacting inappropriately. And if the reaction is strong enough, it can even be life-threatening. For example, somebody with a severe food allergy, that allergy might cause swelling and inflammation in the passageways leading down to our stomach and to our lungs that might be so severe that it would cause death. So these allergies, again, normally the cells that would create a response to an allergen, those would get weeded out during development. However, if they are not weeded out, then an allergy can be strong. And what we're going to see is when we talk a little bit about how the immune system remembers threats, we can also see why an allergy may get more severe with repeated exposure, why a bee sting one day might cause a mild reaction, and a bee sting every day in a row for a month, which sounds horrible and would be, might create a much more dangerous condition. So I hope that today you've gotten a slightly better understanding of how the body's specific immune system functions. In a future video, we'll take a look at antibiotics, our best chemical line of defense against bacterial pathogens. Take care.